All right, everyone, so this is the third video for the digestive system. So we last left off talking about the esophagus and the physiology of swallowing. So now we're going to talk about the stomach. So we'll just briefly go through the anatomy of the stomach. So it's divided into four major regions. So we have at the top where the esophagus meets, this red area, that would be the cardia. All right, so it's kind of around the neck of the stomach. And then this part that kind of bulges over, this is called the fundus. And then the main part of the stomach here is called the body. And then here where it meets with the small intestine is called the pylorus. All right. And so also a couple things to point out that are important would be here, this kind of curve, whoops. All right, this curve here is called the lesser curvature. And then on the other side here, we have the greater curvature. All right, and so the greater curvature is important because that is where the greater omentum attaches. So it attaches to the greater curvature of the stomach and folds over the front of the abdomen and then kind of loops back up and attaches to the transverse colon. All right, so now let's talk about histology of the stomach. All right, so there's a lot of detail for the histology of the stomach. So we're going to take our time going through it. Okay, and so most of that detail is going to come in with the mucosa of the stomach because there's going to be a lot of secretory cells that you're going to need to know what they do. All right, so for the mucosa, it's made up of simple columnar cells with goblet cells mixed in, making a lot of mucus. That mucus is very alkaline in pH, all right, and it forms what's called the mucus or the mucosal barrier, all right. And so the importance of that is, as I'm sure you know, the inside of the stomach, all the juices in the stomach are extremely acidic. And so that alkaline mucus that lines the epithelium helps to protect them against that acidity, okay. And so that's the function of that mucosal barrier, is to protect against the pH. All right, so protects from pH. Okay. All right, and so then if we look at kind of this picture of the mucosa of the stomach, it'll consist of a series of many gastric pits that lead to gastric glands, all right? And so when you're looking down from the surface, you'll see these openings. These are the gastric pits, all right? And then they lead down into these structures, which are the gastric glands, all right? And so within those gastric glands, we have a bunch of different types of cells. So we obviously, as we mentioned, have all of our goblet cells mixed in, making that alkaline mucus for that mucosal barrier. But then as we proceed in, we'll come across first these purple cells. All right, and so all these purple cells are called parietal cells. And so the parietal cells, their function is to secrete HCl. They make that stomach acid, okay? And in addition to that, they have another really important function, which is to make and secrete intrinsic factor. And you need intrinsic factor in order to absorb B12 from foods, all right? The vitamin B12. And you need vitamin B12 for red blood cell production, all right? So it promotes red blood cell production. So somebody maybe who maybe has had like a gastrectomy, part of their stomach removed for whatever reason, they're not making as much intrinsic factor, therefore they're not getting as much vitamin B12, and so they could become anemic because of that, right? And we talked about that type of anemia. Okay, so parietal cells have three types of receptors on them. all three of these receptors, when activated, will stimulate the production of HCl. Okay, so for maximal production of HCl from these parietal cells, you would want to activate all three of these receptors. Okay, and so one receptor is activated by histamine. So when histamine binds, that will cause the cell to release HCl. The other one is activated by gastrin, which is a hormone. And then the last receptor is activated by acetylcholine, the neurotransmitter. Okay, and so as we talk about the physiology of the stomach, we'll go through where each of these signals come from. All right, and so make sure you make a note of that and kind of put those pieces together of activation. Okay, and so then also in the gastric glands, we have these kind of orangey red cells. These are going to be chief cells, and so chief cells secrete the proenzyme pepsinogen. All right, so just a reminder of what a proenzyme is. It's an inactive form of an enzyme. Okay, they're also sometimes called zymogens. That's the same thing. All right, so the reason, and this is very common with enzymes that digest proteins, they're frequently secreted as proenzymes because you don't want those enzymes to just start digesting proteins right here, along the cells that are secreting them. You want it to get out into the lumen of the stomach out here before it starts digesting protein. Okay, so it's important to have those secreted as proenzymes so that they're only activated when they're in the presence of what they're supposed to be digesting. Okay, and so pepsinogen is activated into pepsin, and it's activated by the acidity, right, that HCl within the lumen of the stomach. Okay, so that HCl in the stomach acid, once that pepsinogen gets out of the gastric gland and the pit, right, out here, and it's to that area with all that acid, it will be activated and it can start digesting proteins. All right, and then our last type of cell is going to be a G cell. Okay, and a G cell is a type of cell we're going to see a couple times, called an enteroendocrine cell. These are just the entero part refers to the digestive system, and we know what endocrine means, right? Secreting hormones into the blood. So these are just cells of the digestive system that secrete hormones. All right, and so the hormone secreted by a G cell is going to be gastrin. All right, and so these G cells are activated when food enters the stomach and starts to stretch the stomach. That stretch is going to activate those G cells, which are a type of enteroendocrine cell, and they're going to secrete gastrin. And then, as we just said, right, gastrin's a hormone. It's going to go into the blood, and it's one of the three things that will activate our parietal cells. So that'll stimulate parietal cells to make HCl. All right, and then gastrin also, or sorry, not gastrin, G cells also release histamine, okay, and they release it locally, so in a paracrine fashion, and that will activate parietal cells as well, okay, so two of the signals that can activate those parietal cells are coming from G cells. All right, so that's the mucosa and the cells in the mucosa and what they do. With the submucosa of the stomach, there are folds of the submucosa. All right, so if you imagine there's just like this is actually just in a big fold, right? And then the submucosa down here is in a big fold as well. That is called a rugae. So the submucosa and mucosa are folded. All right, so these are folds of the mucosa and submucosa. Okay, and those rugae just allow for the stomach to be able to stretch, and you can eat like a large amount of food, a big meal, and your stomach will stretch to accommodate that big meal. All right, and then lastly we have, well, not lastly, the third layer is our muscularis externa, and it's unique in the stomach because there is an additional third layer of muscle. So we know our circular and longitudinal layers. Now we have this inner oblique, all right? And it has that extra layer of muscle because the function of the stomach is to really crush up, pulverize, and mechanically digest that food. So that extra layer of muscle going in a third direction allows for it to better grind up the food. All right, and so then also the stomach is intraperitoneal, so it would have serosa. All right there. Okay, so let's talk about the physiology of the stomach. So the function of the stomach is to take that bolus that came from the mouth, right, that chewed up food mixed with saliva, and we're going to convert that into chyme. So chyme is like a milky white substance, right, of partially digested food, right? It's just a milky white fluid. Okay, so we're going to convert that bolus into chyme. And so that's going to happen, like we said, through mechanical digestion. That's with those 3 layers of the muscularis externa, and through chemical digestion. All right, and so we just mentioned the stomach, those parietal cells are secreting a lot of pepsin, a lot of HCl. All right, the chief cells are making that pepsin. And then also don't forget inside that bolus, right, there's still going to be some salivary amylase, a little bit of residual salivary amylase digesting carbohydrates, and that will keep digesting until that bolus is fully broken down and the HCl in the stomach denatures that enzyme. Okay, so it'll function until it's denatured by the acidity of the stomach. All right. So then there's 3 phases for gastric secretion, right? The release of, all these components, particularly this HCl, all right? There's 3 phases for that. So let's go through those. So the first phase is called the cephalic phase, all right? And so we know cephalic means your head. And so this does pertain to your brain. And so this stage only lasts for a few minutes. And it occurs when you either see, smell, or even think about food, all right? And so the brain sends a message through the vagus nerve, which will release acetylcholine onto parietal cells. And we know acetylcholine is one of the things that will bind to the receptors on parietal cells and cause it to release HCl, okay? And you also know, I'm sure, just from your own experience, that smelling something that smells really good and tasty will make you salivate, right? That's also part of the cephalic phase. So salivary glands are activated in this phase as well. Okay. And so then our second phase lasts about 3 to 4 hours. It's called the gastric phase. Gastric meaning stomach, so this is once that bolus of food reaches the stomach. All right, so once it enters the stomach, that's going to stretch the stomach out a little bit. All right, and that stretch, there are going to be stretch receptors that are going to be acting in 2 different ways. All right, so they'll act through the enteric nervous system, right, that will cause the stomach to start churning, and it'll also send a message all the way up to the medulla, which again, kind of like in our cephalic phase, will act through the vagus nerve to release acetylcholine, which will activate our parietal cells. Okay, so those receptors for stretch will stimulate the stomach to start churning and grinding up that food, as well as releasing acid to start breaking it down. Okay, and then there are also chemoreceptors in the stomach, and the chemicals they are detecting is going to be an increase in pH. So the acidity of the stomach is usually very, very low, around 2 or 3. And so when you add, food, a bolus of food, that's naturally going to have a higher pH, and that's going to bring the pH up. So these chemoreceptors are going to detect that change in pH, as well as the presence of peptides, which makes sense because the only enzyme made in the stomach is that pepsin, which digests protein. All right, so it's going to be on the lookout for that protein as well. Okay, so when these chemoreceptors are activated, that is what is going to activate our G cells. Remember, our enteroendocrine cells in the stomach. Those G cells are going to release gastrin and histamine that both act on parietal cells, causing them to release HCl. Okay, so both our cephalic and our gastric phase are activating those parietal cells. Okay, and then our third phase is the intestinal phase. In the intestinal phase, right, food is now leaving, that chyme, right, is now leaving the stomach and entering the small intestine. So the first segment of the small intestine is called the duodenum or the duodenum. And so when food enters the duodenum, that will tell the stomach that its job is done, right? It's converted that bolus into chyme, because only once that bolus has been converted to chyme can it actually pass into the duodenum. And so the stomach will stop its secreting and its churning actions at that point. Right? This occurs through both a neural component and an endocrine component. So the neural component comes from the duodenum being stretched by the addition of that chyme. All right, that's going to activate the sympathetic nervous system, which counteracts digestive activity. So that sympathetic nervous system is going to shut down the secretion and churning of the stomach. Okay, and then also in the duodenum, there are duodenal enteroendocrine cells, right? Just like we talked about the G cells in the stomach, there are endocrine cells in the duodenum as well. And they secrete two different hormones. All right, and we'll talk about where they are when we do the histology of the small intestine. But they're activated by a few different signals. So they're activated, one, by the low pH of that material coming from the stomach, right? It's very acidic. That acidity will activate these cells, as well as the presence of fats and carbohydrates, because that is one, both of these things are going to be digested in the duodenum. So it's going to be detecting for the presence of those. Okay, so the hormone released in response to those fats and carbohydrates is CCK. All right, and so CCK is a hormone that has a few different functions. All right, and so one thing it does is it, as our neural component did as well, is stops the churning of the stomach and the production of those juices. It also causes the gallbladder to contract, all right? And that's really important because, and we'll talk about with the liver, what exactly bile does, but bile is needed for the digestion of these fats, all right? It has a really important function in that. All right, and then also CCK will stimulate the pancreas, and the pancreas will secrete in response to CCK a pancreatic juice that is really rich in enzymes, and so especially enzymes that are going to digest carbohydrates and fats. But really, the pancreatic juice has enzymes to digest all of our macromolecules. Okay, and then lastly, the CCK tells the hypothalamus, signals that you're full. Okay, and so that's when you start to feel full, which is interesting because that's a while after you actually ate, right? We've already gotten through that cephalic phase, the gastric phase, and we're in the intestinal phase, and we're signaling now that you're full, all right? That's why you can eat such a large meal, especially if you eat really quickly, and then later you'll feel miserable because of this CCK, right? Sending that signal you are really, really full. Okay. And then our second hormone is called secretin. So secretin is the hormone released in response to that low pH coming from the stomach. And so it is also going to shut down stomach activity. as well as signaling the pancreas. However, instead of signaling the pancreas to release enzymes, it signals the pancreas to release a really alkaline pancreatic juice, right, with a lot of buffers in it. All right, it's going to have a lot of bicarbonate and stuff like that in it to help neutralize, that's the word I was looking for, neutralize that pH, right? Bring it back up. So that duodenum and the other parts of the small intestine, as that chyme moves through, is not going to be damaged by that acidity. Okay? All right, so that is it for the stomach, right? And so how the stomach is activated, the different signals, and then these phases of kind of stomach physiology and what happens when you eat. All right, so now let's move on to the small intestine. So first we'll talk about anatomy. So the small intestine is divided into 3 different regions, and they each are kind of shown here on this picture. So the first region that is connected to the stomach is the duodenum or the duodenum. I honestly switch back and forth between pronunciations. I don't know why, but I always have. So that's the first segment here, all right? And that's what we were just talking about. That's what's going to be signaling the pancreas and the gallbladder and neutralizing, that acidity, all right? So it's going to receive that chyme directly from the stomach. So this is where, because the pancreas is going to be secreting its juices, as well as the liver, its bile directly into the duodenum, that is where carbohydrate and fat digestion is going to occur. All right? And so then the next segment of the small intestine is going to be this purple area. All right? That is the jejunum. And so the jejunum is going to be the area, and I've seen different drawings where the jejunum takes up a lot more, and even some with a lot less. Typically, I like to think it's the majority, right? If I were to actually draw this myself, maybe adjust the colors a little bit, I would have all of this being purple, because the majority of the small intestine, right, the actual length of the small intestine is going to be jejunum. And that's where absorption takes place. So that's absorption of nutrients. Whoops. All right, so our carbohydrates, our fats, our proteins, our nucleic acids, vitamins, minerals, and especially water, right? All of that is absorbed in the jejunum. All right, and then the last segment, all right, and this is the segment that's connected to the large intestine, is the ileum. Okay, and so the ileum, when we look at the histology, is going to have a lot of those malt tissues, right? Those lymphoid nodules, because more and more bacteria is going to be growing as that food is moving through these intestines, right? So by the time it gets here, and then especially in the large intestine, there's going to be a lot of bacteria, so that lymphoid tissue is going to help combat that bacteria. So as you move through the small intestine, histology-wise, you'll see more and more and more of those lymphoid nodules the further you get and the closer you get to the large intestine. Okay, so let's talk about histology now. All right, so kind of like with the stomach, the majority of the interesting stuff with the small intestine is going to be in the mucosa, and then there's some structures in the submucosa as well that you should be aware of. So let's talk about the mucosa first. So the mucosa for the small intestine contains simple columnar with goblet cells, and those simple columnar cells are going to have microvilli. Okay, and so the microvilli are these extensions of plasma membrane. All right, so that's on one single goblet cell. They have little extensions of plasma membrane. Those are called microvilli. And their function is to increase surface area. So we said small intestine is where a lot of absorption occurs, so increasing that surface area is extremely beneficial. All right, that's going to be kind of a theme with the small intestine. However, also these microvilli make up what is called the brush border. Okay, and so the microvilli have embedded in them some digestive enzymes, right? So if I were to draw this, I would draw kind of these little, if we zoom in on this guy, right? A bunch of little enzymes dotting all around here, okay? And so then as your small intestine contracts, right, to kind of squeeze, whether it's segmentation to kind of, churn that food around, and actually one function of segmentation is to press that material against this brush border so it comes into contact with these enzymes, all right? And so as it does that, right, those nutrients will come in contact with these enzymes, and their function is they're kind of the last stage of digestion. So most of the nutrients have been digested at this point, and they're down to kind of 2 monomer units. All right, so like a disaccharide or a dipeptide, right? So 2 amino acids together, or maybe 2 glucoses stuck together. And the job of these enzymes is to break that last bond so that now the monomers can be absorbed. Okay, so let's talk about some of those enzymes. All right, so if we're talking about our disaccharides, right, our last residual carbohydrates, they could be the disaccharides we talked about: maltose, lactose, and sucrose. So the enzymes for those that would be here in this brush border would be maltase, lactase, and sucrase, all right, which is nice, very similar names. And then for our peptides, right, we have the enzyme dipeptidase, so this would be for a dipeptide, all right, and then we also have the enzyme aminopeptidase, okay, and so typically with peptides, these enzymes are going to target very specific bonds between certain types of amino acids. All right, so there's going to be a lot more than just this, but this is just the ones that you need to know as examples of brush border enzymes. Okay, so in addition to having these microvilli, right, that add surface area, these folds on the cell membrane, the mucosa itself is in these large folds, and those make up a villus, or if we're saying plural, it would be villi. All right, and so this would be one villus here, right? So there's these kind of finger-like projections of the mucosa, right? So the mucosa only. So the submucosa is not folded with it, just the mucosa. Okay, and so within these villi are going to be these lymphatic structures called lacteals. All right, and we mentioned those before. So lacteals are these lymphatic vessels located in the villi, and they are going to be really important for fat absorption. Okay, so that's one thing with the villus. It contains these lacteals for fat absorption. Also, it is going to increase surface area as well. All right, surface area. So if you think about it, if this mucosa were just straight across, so from here, right, to here, we just had a straight chain of columnar cells, think about how many fewer columnar cells you'd be able to fit in there compared to what we have here now that we have this big villus, right? Many, many more columnar cells. That's what it means by increasing the surface area, right? You have all those cells, and then on each one of those cells you have all of these microvilli, which is just space for nutrients to be absorbed. Okay, so in addition to these villi, between the villi, the mucosa kind of dips down like this, and actually, especially when you're looking at a slide in lab, right, one way to identify these are called crypts of Lieberkühn, right? I'm just going to label it here, call it a crypt, so I don't have to take the time writing out Lieberkühn. All right, so this is a crypt. Also, sometimes I think in textbooks now they're starting to call them intestinal crypts. or intestinal glands, maybe, I'm not sure which one it is, but any of those are acceptable on a test, because they've changed a lot of names lately. They're taking away a lot of these names of the people that researched and found them and replacing them with more generic names. But anyway, so these crypts, when you're looking at a microscope slide, a way you can identify them is that essentially these little pockets of columnar cells basically touch the muscularis mucosa here. All right, so they dip down all the way to the muscularis mucosa, and then in these crypts, we're going to have some secretory cells. All right, in particular, we're going to have these enteroendocrine cells. All right, and these are the same cells we talked about with that intestinal phase. All right, these are the cells that are releasing that CCK and secretin during digestion. Okay. All right, so the crypts of Lieberkühn contain these enteroendocrine cells, and they also contain Paneth cells. And so Paneth cells are going to secrete lysozyme, which is going to help, it's an antibiotic, help reduce bacterial growth, and defensins, which will also protect against, infections and pathogens. Okay? All right, so that's coming from these crypts, so they would just secrete them up into the lumen of the small intestine. All right, so that's the mucosa. So now let's talk about a couple of important features of the submucosa. So with the submucosa, it is also going to be folded. So you can see here, right, these would be villi, so just folds of the mucosa. And then the submucosa as well is going to fold. All right, and so these folds are called placae circularis. All right, and so that name just comes from the fact that they're circular folds, all right, meaning that if we're thinking of, the small intestine, right, as kind of a tube like this, right, the placae circularis are these folds, these ridges running in kind of circles down the length like this, right, instead of being longitudinal, which means the folds would run this way. That's what we saw in the esophagus, right? So with the small intestine, they're kind of encircled like that. Okay, so the placae circularis, that also will increase surface area even further. And then in the small intestine, we have two important structures. So one, the first one, all right, here, these are going to be Brunner's glands. And so you find them in the duodenum. Okay, so these are, Brunner's glands. And so their job is to help neutralize that acidity coming from the stomach. So they're going to secrete a bicarbonate-rich mucus to help protect the duodenum from that stomach acid. Okay, and so then our second structure we can see here, this is one of those lymphoid nodules that I mentioned. Okay, so this is called a Peyer's patch, typically found most abundantly in the ileum. All right, and so with the Peyer's patches, they look like these big, a lot of times they're huge, right? They're these giant masses of white blood cells. And frequently the majority will be in the submucosa, but you'll see that they've invaded into the mucosa as well. So a lot of times when you're looking at an actual slide, where these Peyer's patches are, it'll be kind of obscured as to where the submucosa stops and the mucosa starts, because it'll just be full of white blood cells. All right, so these Peyer's patches, and they're going to help prevent bacteria that are in the lumen out here from getting into this blood supply here that's in the submucosa. Okay, and then we have our muscularis externa. It just has two layers, right? That inner circular layer and the outer longitudinal layer. Their function is in peristalsis, all right, and segmentation. So both of those are going to be happening. Remember, again, segmentation is uncoordinated contraction, and that is going to be to mechanically digest and also to help that food material come in contact with that brush border. And then we have peristalsis that's going to move that material forward through the intestines. Okay, and so then we're going to have with the small intestine a mix of adventitia and serosa. The duodenum is retroperitoneal, so it would have adventitia. And then the rest of the small intestine, the jejunum and the ileum, are going to be intraperitoneal, so they're going to have serosa. All right, so those are our alimentary canal organs so far. We just have the large intestine left to talk about in a little bit, but we need to go back and talk about the accessory organs that were involved here with the intestines. And so that's going to include the liver and the gallbladder, which we'll talk about right now. And then in the next video, we'll discuss the pancreas and the enzyme secreted by it and its function. Okay, so the liver. All right, and so the liver has many, many different functions. All right, it functions in storing nutrients. It obviously makes bile. Those are important for digestion, but it has many functions in red blood cell recycling and processing and many other things that it does, detoxifying blood, that kind of thing. Okay, so first we are going to talk about the histology of the liver. All right, and so the liver, when we look at the liver on a slide, so one, it's intraperitoneal, so it's going to have serosa, all right, around it, a serous membrane covering it. And then when you look at a slice of the liver, what you're going to see is it's divided into these lobules, all right, so these are these hexagon-shaped structures here, right, so that would be one lobule there. And in the middle of each lobule is this kind of circular opening that's called a central vein. Okay, and so within each lobule, these are basically just these large clusters. So all this kind of teal area is going to be hepatocytes, right? So these big clusters of hepatocytes, and then we're going to have kind of these spaces in between them, all right, that are going to be called sinusoids. Okay, so we'll talk about that in a minute. But before we get to that, let's talk about what's between these lobules. Okay, and so if you look, you see these regions between lobules where you have these green, blue, and red structures. Okay, so these are called the triad region. Okay, so they're found between lobules and they contain three really important structures. And so these three structures are one, a branch of the hepatic artery. All right, so this is going to be bringing oxygenated blood to the liver, all right, to provide it nutrients and nourishment to all those liver cells. And then the hepatic portal vein, which is bringing the blood from the intestines where all that nutrients have been absorbed, right? So this is going to be nutrient rich blood from the intestines. And the job of one of the jobs of the liver is going to be to process those nutrients. So the liver is going to, if there's a lot of carbohydrates, it's going to take that and make glycogen, and it's going to, sort out the different things, store fats, and send the nutrients on to wherever they need to go. Right? And then the third structure is going to be a branch of the bile duct. Okay, and so we know one function of the liver is to make bile, and so that bile is sent out of the liver through the bile duct. Okay, so if we zoom in on kind of one little segment here of the liver, all right, that's what we're looking at here. Okay, so we're zoomed in so we can see the cells, and we can see, so here's our artery, right, here's our portal vein, and here's our bile duct. All right, and so then these cells here, right, all these kind of teal cells, these are going to be our hepatocytes. And then between them, these purple areas are going to be called sinusoids. Okay, and so we know sinusoids, we talked about capillaries and different types of capillaries, right? They are very porous with fenestrations and big gaps. And so their job is they're going to carry blood from this hepatic portal vein, right, this branch of the hepatic portal vein here. That blood is going to flow into the sinusoid, right? And it's going to move towards the central vein, okay? And so that is where, so this blood moving through is going to come in contact with these hepatocytes, and they're going to process all those nutrients like we talked about. All right, and then we have the blood here from this hepatic artery that is actually bringing oxygenation to these hepatocytes themselves. And so that's going to travel through here, and it's going to mix, and so it kind of becomes a purpley color. We're mixing that oxygenated and deoxygenated blood. Okay, and so within these sinusoids, there are also these cells. These are macrophages, and they're called Kupffer cells. And so they just hang out in the sinusoids and help to remove debris and that kind of thing from the blood that's moving through there. All right, so we know we have blood moving through the sinusoids, but then also the liver is making bile. And so the same cells, these hepatocytes, also make bile. And so that bile is going to be secreted into these little tiny green kind of rivers. They're called bile canaliculi. And that's going to be carried to the bile duct here. And then that'll be carried out of the liver. All right, so these are the bile canaliculi here, right? That would be a bile canaliculus. Okay, and so the hepatocytes are going to make the bile, and then that's going to travel through that bile canaliculus to a bile duct. All right, so let's talk about what bile is and what it does, why it's important. All right, so the composition of bile contains bilirubin. All right, and bilirubin is a pigment from the breakdown of heme. Okay, so when we break down hemoglobin, that heme can be broken down further into bilirubin, and that is a bile pigment. All right, so bilirubin is found in bile. Bile is also going to contain some water and ions as well as cholesterol. And then really important for its digestive function is that it contains bile salts. Okay, and bile salts are amphiphilic molecules. And so that just means that they have a polar and a non-polar region. All right, so they have kind of this polar head and then this non-polar tail. All right, very similar to phospholipids that you learned about last semester. Right, those are amphiphilic as well. And so what these bile salts can do is they help to emulsify fat, right? And that means that they take these big fat blobs, right, and they help to break them down into small fat blobs, right? And then they can be surrounded by these bile salts. And so they're surrounded in such a way that because fats, right, are nonpolar, they don't like to dissolve in water and any kind of fluid. All right, and so these bile salts are going to help them to dissolve in that fluid so they can be transported. Okay, and so what they'll do is they'll face their nonpolar part, all right, that's going to like those fats. They'll face it towards the fat. And then they'll leave the polar part out facing the watery environment. Okay, and so by doing that, they're going to kind of shield, put like a force field around those fats and protect them and allow them to move through that watery environment. Okay, and so then emulsifying the fats. So one other important function of that is taking this big blob of fat and making them into these smaller droplets. All right, and so that's important because the enzymes that are going to break down the fats would have a really hard time kind of chewing through these outer layer of fats and getting to all the fats on the inside. So these smaller droplets are much easier for the enzymes to work with as well. Okay, so let's talk about the pathway of bile. All right, so once it's made, all right, what path does it take? Okay, so we said it's made by the hepatocytes in the liver, travels through those bile canaliculi to a branch of a bile duct, and so then that is going to travel through these left and right hepatic arteries. All right, so this is left and right hepatic artery. Those two are going to join to form the left and right hepatic duct. They're going to join to form the common hepatic duct. All right, so left and right join to form a common hepatic duct. And then from there, the bile can either travel into the gallbladder, and so it will do that by going through this structure here, which is the cystic duct, or it can travel downwards, right, and be added straight into the duodenum by traveling through the common bile duct. Okay, and so then that would lead to the duodenum. We'll actually see in the next video that common bile duct would join up with the pancreatic duct, right, and they both together enter the duodenum. Okay, so you should know that pathway for bile. And so I would recommend, like the way I learned it was actually just drawing this, right? So I would just draw, like we have our left and right hepatic ducts. They're going to join to form the common hepatic duct. And then that'll branch off to the cystic duct here for the gallbladder, and it continues down to the common bile duct, right? And then that will leave, we'll see a little later, and join with the pancreatic duct and go to the duodenum. So if you just practice drawing it out and doing it like a story like that, eventually it'll become kind of more natural and much easier to remember. Okay, and so then the last topic for this video is going to be the gallbladder. All right, and so the function of the gallbladder, and we can see it here, right? It's this kind of pouch here. It sits on the posterior side of the liver, and its histology, it's just made up of, it's very simple. It's simple columnar, smooth muscle because that gallbladder can contract to release the bile, and then it's surrounded by serosa. Okay? And its function is to store the bile and to concentrate it. So by concentrate it, we just mean that it's going to be removing water. Okay? So it's going to remove water to concentrate that bile. So then when that gallbladder contracts, the duodenum is going to get this really concentrated kind of shot of bile. Okay? And so gallstones, that should have been up here. All right, so remove water. And so gallstones can form, and if it's chronic, a lot of times the gallbladder will be removed. All right, and they form because the gallbladder perhaps over-concentrates the bile. And so that bile, so the cholesterol and the salts in the bile will start to crystallize, and they'll form these gallstones. And so they can be very painful to pass if you are able to even pass them, all right? Because imagine they have to travel through all these ducts, right? So they can get stuck in that cystic duct, they can get stuck in this common bile duct, or even when this joins that pancreatic duct, right? They can get stuck on the way to the duodenum. And so that whole way, and generally these gallstones are not smooth on their surface, so that whole way they're kind of abrading and, damaging that tissue along the way, which is very painful. Okay? All right, so that's it for this video. So we'll pick up next time and talk about the pancreas, and then finish out the rest of the digestive system, as well as how nutrients are absorbed in the small intestine.